Lecture for Chapter 7, Control of Microbial Growth, Part 1. In order to understand how to control microbial growth, it's important to review what is required for microbial growth in the first place. All vegetative cells need to have a source of nutrients, and those nutrients will be used in various metabolic processes. Some of those metabolic processes involve catabolic reactions that will release enzymes and break down those large molecules into smaller molecules. Then that energy and those smaller molecules will be used in anabolic reactions to produce larger molecules, such as the macromolecules, which will be used to form cellular structures and also help the cell perform important cellular processes. And important to all of this metabolism are the enzymes. And it's very important for the cells to have functional enzymes. If the enzymes are not functional, then the cell cannot metabolize, and if the cell cannot metabolize, it cannot survive. Going into more detail about the requirements for microbial growth involves reviewing information from previous lectures. So as I mentioned in the previous slide, all microbes need a source of nutrients, and those nutrients are the chemical requirements for growth. And they need macronutrients, which are atoms in relatively high concentrations so that they can build up their macromolecules. They also need trace elements and growth factors. Trace elements act as the cofactors. Growth factors act as the coenzymes, which are needed to make the enzymes functional. And of course, you need functional enzymes to facilitate metabolism. Speaking of the enzymes, that relates to gene expression. Remember, gene expression is where the information to form the proteins is in the form of DNA. That DNA gets transcribed into RNA, and the RNA is translated into the appropriate protein. And most of the proteins that a cell produces are enzymes. So in order to have the correct enzymes, you need to have the correct genes, so that they can be transcribed correctly, then translated, and then you get the correct polypeptide. Gene expression is not the only thing that is needed to get functional proteins. Also, protein structure is important. So that primary structure that relates directly to the sequence of amino acids, which is encoded by the DNA. But once you have that primary structure, then those amino acids have to be able to hydrogen bond to each other to form the initial secondary structures, the alpha helix and the beta pleated sheet. Once you have that secondary structure, then the polypeptide will form its tertiary structure, which also involves hydrogen bonding. And once you have the three-dimensional structure, that tertiary structure, then that polypeptide interacting with other polypeptides will give you the quaternary structure, which is the functional enzyme or the functional protein. So once a cell has the nutrients to use in metabolism and has functional proteins, then it's important to have the physical requirements of growth to maintain the activity of those enzymes. Temperature is an important aspect of the physical requirements for growth. So for the enzymes to be functional, they must be at their optimal temperature to function at their highest ability, or at least within their temperature range. Same thing for pH. Same thing for pH. Enzymes have an optimal pH. If you change that pH, that will cause the enzymes to denature, and then they will be non-functional. The last physical requirement dealt with osmotic pressure. And osmotic pressure basically relates to the environment that the bacteria will be put into. If that bacterium is put into an isotonic environment or a hypotonic environment, the bacterium should be functional. So isotonic environments, no osmosis will occur. Hypotonic environments, as long as they have an intact cell wall, the bacterium should be able to survive. Hypertonic environments will kill the bacteria, and it will kill the bacteria because osmosis will occur, water will move out of the cell to the point where plasmolysis will occur. The plasma membrane will shrink, and the cell does not have enough water to maintain the secondary and tertiary structure, the hydrogen bonding for the proteins, and also it does not have enough water for catabolic and anabolic reactions. Remember that those catabolic and anabolic reactions both involve water molecules. While the previous chapter was all about promoting microbial growth, getting the microbes to grow, this chapter is about controlling microbial growth. And when we talk about controlling microbial growth, we're talking about preventing the microbes or the bacteria from growing. And there are several instances where this is very important. One is in this class. When we go through the lab portion of the class, we are interested in getting bacteria to grow, but only the bacteria we want to grow. So it's very important in the experiments that we control the growth of unwanted bacteria. And a couple of ways we do that is by disinfecting the bench and using the Bunsen burner to sterilize the loop and other equipment. In a medical setting, it's very important to control the microbial growth in the medical facility so you don't transmit an infection to another patient or to the healthcare workers. And also, when a patient comes in with an infection, it's important to control the microbial growth within that patient's body, basically to stop the microbial growth in that patient's body so that we can treat the infection and cure the patient. The last example of where it's very important to control microbial growth is something that affects all of us pretty much every day, and that's with food. When food has spoiled, what that means is that microbes have colonized the food, they're able to use the food as nutrients, and then they are growing, increasing their numbers on the food. And not only are they increasing their numbers, but they are also releasing waste products from that metabolism. And so the increasing numbers of microbes and the waste products makes the food inedible for us. And so we say it has spoiled. Before we get into the different methods of microbial control, it's very important to go through some vocabulary terms. And a lot of these terms are often used interchangeably in regular conversation, but in a medical or laboratory situation, it's important to know the different definitions of these words and to use them correctly. The first term is sterilization. Sterilization is basically defined as the removal or destruction of all microbes in any form. So if something has been sterilized, that means there are absolutely no microbes in any form on them. Surgical equipment is the first example of things that are actually sterilized. So surgical equipment or dental hygiene equipment, this needs to be sterilized before it's used on the patient. So absolutely no microbes should be present on this before it's used. A second example is equipment in a laboratory. So all the media we use is sterilized. And also before you use the loop, you need to sterilize it by flaming it in the Bunsen burner. So in a laboratory setting, there are things that are sterilized. Last example of sterilization is commercial sterilization, and this is used to process food. So especially canned food, it's important to sterilize the food and to kill the endospores. So if you remember with bacteria, some bacteria like Bacillus and Clostridium can form endospores that are highly resistant to forms of sterilization and disinfection. And the reason you need to kill these endospores is they can cause diseases such as botulism. The next term is disinfection. Disinfection is basically a step down from sterilization. And if something is disinfected, that means all vegetative microbes are gone, but endospores may still remain. So to disinfect something, often disinfectants are used. And a disinfectant is an agent that has the ability to kill all the vegetative microbes, but it has to be used correctly in order to do so. So often to disinfect something, A disinfectant needs to be used, but it has to have a high enough concentration, and it has to be left on the surface long enough to work to kill all the vegetative microbes. So in reality, most things are not disinfected. So in reality, when we use disinfectants, the end result is that the surfaces are most likely sanitized. And sanitization is defined as reducing the number of microbes to safe levels. So if something is sanitized, it still has microbes present, but they've been reduced to safe levels. So when you are using disinfectants to clean your kitchen or bathroom, you are probably not truly disinfecting those surfaces. You're not removing all the potential vegetative microbes, but you are reducing them to a safe level. And a lot of equipment, like equipment in a meat packing plant, will also be sanitized because it's almost impossible to truly disinfect these surfaces or these types of equipment. Antisepsis is the term used when a disinfectant is applied to living tissue. And of course, it is a disinfectant, so it does have the ability to kill all the vegetative microbes on the skin, but in reality, this is probably not going to be accomplished. But it should sanitize the skin, it should reduce the microbes to safe levels to prevent infection after the surgical procedure. Degerming is the specific term we use when an alcohol swab is applied to the skin before an injection. And the purpose of the alcohol swab is for the alcohol to kill microbes and for the swabbing action to physically remove some microbes from the injection site. Ideally, this would be nice if it was actually disinfecting the skin, but in reality, it is probably just reducing the number of microbes in the area to prevent infection. The last two terms I want to go through are bactericidal and bacteriostatic. And the first one, bactericidal, bacterio refers to bacteria, cidal means to kill. So this refers to a treatment that will result in the death of the bacterial cells. And there are some terms that relate to this, germicide or germicidal. Germ refers to microbes, so a germicidal is a treatment that will kill microbes. Fungicide, fungicidal, it's a treatment that kills fungi. The other term, bacteriostatic, again, this refers to bacteria, and static relates to stasis, staying in one place. So if something is bacteriostatic, it prevents the cells from dividing. So the bacterial cells are still alive, they're still metabolizing, but they cannot replicate. So the number of cells will not increase. So some control agents, some control treatments are bactericidal, which will outright kill the bacteria. Others are bacteriostatic, which just prevents the bacteria from replicating. When determining what control method to use, there are a couple factors to consider. And the first factor to consider is the actual number of microbes present. And just logically, if there are a lot of microbes present, then the control method that you are going to use needs to be stronger or longer. Compared to an area that has very few microbes present, then your control method does not have to be as strong or aggressive. The second factor to consider are the individual characteristics of the microbes that are contaminating the surface or the liquid. The first division is between gram-positive bacteria and gram-negative bacteria. And in general, gram-negative bacteria tend to be more resistant to control methods. And that is because of the structure of their bacterial cell wall. Remember that the gram-negative bacteria have that outer membrane that can prevent chemicals, antibiotics, other molecules from going into the cell. So in general, gram-negative bacteria are more resistant. So if they are present as a contamination on the surface, your control method will need to be stronger than if it was just gram-positive bacteria. Another bacterial characteristic is the ability to produce endospores. Remember, endospores are highly resistant to all forms of disinfection and sterilization. So if you have endospores present on the surface, you're going to need a much more aggressive control method. The last factor to consider is the presence of organic material. Organic material tends to reduce the effectiveness of the different control methods. So if there is a lot of organic material present, the control method is going to have to be stronger or longer in order to sanitize, disinfect, or sterilize that surface or liquid. One example is sewage. Sewage has a lot of fecal material in it, and so to treat sewage, you're going to have a longer, stronger treatment than if you were treating water in a swimming pool, for instance. Also, bodily fluids contain a lot of organic material, so things like blood, pus, vomit, diarrhea. If that is contaminating a surface, your treatment, your control method is going to have to be stronger than if there was no organic material present. Even though there are various methods to control microbial growth, of all these methods, there are only a few modes of action they may have. Mode of action is basically the target, how they actually inhibit the growth of the microbe or kill the microbe. And the first mode of action or target are membranes. So remember, the membrane serves as the selectively permeable barrier for the cell. So it separates the inside of the cell from the environment. Also, the membrane is selectively permeable, so it controls nutrients going in and waste products going out. So if this membrane is disrupted in any way, you can destroy the ability of the cell to transport nutrients in and waste products out, and that could kill the cell, or you could disrupt the membrane completely, which would destroy the structure of the cell, killing the cell. The second type of mode of action is to have an effect on the macromolecules. And the two most important macromolecules are the proteins and the DNA. And from previous chapters, you should already know the importance of the proteins and the DNA. Proteins, vast majority of the proteins serve as enzymes. So anything that can alter the structure of the enzyme or prevent enzymes from being produced will have an effect on the metabolism of the cell. And if the cell can't metabolize, it can't survive. DNA, the importance of the DNA is it serves as the molecule of information, and it holds the information to make the enzymes. So if the DNA is damaged in any way, if it's destroyed, or if it's mutated, that inhibits the cell's ability to produce the enzymes, which again inhibits the cell's ability to metabolize, and if it can't metabolize, it will die. So as a review from the previous chapters, in order for a bacterial cell to be able to metabolize and grow and divide, it needs chemical requirements, it needs gene expression, it needs the proper structure of the enzymes, it needs its physical requirements such as temperature, pH, and osmotic pressure. So if you don't want the microbes to grow, if you want to control their growth, prevent them from growing, you can affect any of these things. You cannot allow them to have the nutrients that they need, the chemical requirements for growth. You can damage the DNA or prevent the process of transcription or translation. You can alter hydrogen bonding, which will have an effect on the tertiary structure of the protein and which will lead to non-functional proteins. Or you can take the bacteria out of their temperature range, out of their pH range, or you can put them in a hypertonic environment. Before going into the details of the different methods of microbial control, I just want to go through this list of microbes that organizes them by their relative resistance. And this involves reviewing the basic structure of the microbes. So first, going through the most resistant microbe, the one that is the most difficult to kill or inactivate, are the prions. And remember, prions are infectious proteins. And some scientists believe that actually there is no way to sterilize surgical equipment that has been exposed to prions. So that's under discussion, but it is possible that once surgical equipment has been exposed to prions, it cannot be sterilized. You can't get rid of that. Going down, if you compare the endospores of bacteria to the different types of bacteria, endospores are obviously more resistant. And they're more resistant because, remember, they are the protective structures of certain types of bacteria, the Clostridium and the Bacillus. So they should be more resistant than the gram-negatives and the gram-positives. Looking at the bacteria, gram-negatives are more resistant than gram-positives. And I went through that before because the gram-negatives have that outer membrane, so it can prevent chemicals from getting into the cell and having those negative effects. Mycobacteria, I've mentioned them before. They are very resistant. These are the Mycobacteria leprae, Mycobacteria tuberculosis. They are acid-fast because they don't have very much peptidoglycan, but they have a lot of mycolic acid and waxes in their cell walls. So that makes them very resistant. They're actually very difficult to kill. Talking about the protozoa, two forms of the protozoa, the cysts of the protozoa and then the vegetative protozoa. Remember, they are referred to as trophozoites. The cyst, the whole purpose of forming a cyst is to protect the protozoa. So the cyst is the protective form, so it should be more resistant to disinfection, sanitization techniques. Then you have the fungi here. So the fungi are just about in the middle. But the last two, the viruses, viruses without envelopes, those are the naked viruses, and viruses with envelopes. And viruses with envelopes, lipids, it's very easy to disrupt lipids. And remember, the whole purpose of the outer covering of the virus is that is how the virus recognizes and attaches to the host cell. So if the envelope is disrupted, the virus has been inactivated. It has no way to recognize and attach to the host cell. So it is much easier to disrupt a lipid bilayer, an envelope, than a capsid. Remember, the naked viruses, their outer surface was a capsid. So this is a list of microbes, general groups of microbes, from most resistant to least resistant. Least resistant, easiest to kill or inactivate. So you should review the structures of the different microbes and how that relates to their ability to resist these different methods of control.